right. Before I start, I'd like to thank organizer Professor Che and the chairman Professor Lee. I'm really honored and pleased to join this forum and share our results about the cell therapy. Today, my title is Personalized Cell Therapy for Parkinson's Disease. Is it hope or reality? So my research has been the dopaminergic system and Parkinson's disease. I realized that time really flies so fast because Dr. Lee introduced me that I have been doing this dopaminergic research for last 25 years. But when I counted, actually, now I realized that it's 33 years. So really time flies. But anyway, when I joined, 33 years ago, Cornell Medical School as a junior faculty, I started my research in the dopaminergic system. And dopamine is a important neurotransmitter, and it is a very critical research area, which was pioneered by Swedish scientist, the late Professor Arvid Carlsson, who identified the dopamine as an important neurotransmitter. And for that work, he got the Nobel Prize in the year 2000. And when I checked 2 days ago in the PubMed, I checked dopamine, and I found that there are papers about dopamine in 171,000 papers so far. So I really, was impressed that by one finding, the whole scientific field has opened and doing a lot of research. As a very short introduction, because I know that some of you are not in this field, dopamine is in our brain synthesized by two steps, the two enzymes, tyrosine hydroxylase and the dopa decarboxylase. And the tyrosine hydroxylase is a very famous protein in the neuroscience field because it is the first and the rate limiting step for the dopamine biosynthesis, and it converts L-tyrosine to L-dopa, and then L-dopa is converted to dopamine by the enzyme dopa decarboxylase. Oops, sorry. And actually, once after Dr. Carlsson found that dopamine is a very important neurotransmitter, and then subsequent works found that the dopamine is also critical for our movement control. And then when dopamine synthesizing brain area in the substantia nigra, the midbrain is degenerating, dying, then the patients become the Parkinson's patients. And actually, until this now, the second, the intermediate, the molecule L-dopa is the most standard treatment for the Parkinson's patients. So briefly, in United States only, there are at least 1 million to 1.5 million patients suffering from Parkinson's disease. And each year, there is about 60 new patients diagnosed of Parkinson's disease. And the economic burden is very big. But the important thing is that because of the aging population, the number of the patients are increasing, and the economic burden is also increasing, and they predict that the cost will become double or triple by 2040. So in summary, age-dependent and selective degeneration of dopamine neurons in the substantia nigra is the pathological hallmark of Parkinson's disease. And right now, the major treatment for Parkinson's disease is a pharmacological replacement using L-DOPA or dopamine agonist. But these treatments usually result in a very severe side effect, such as dyskinesia. And so there is currently no treatment that can slow down or stop the disease progression. So my lab has been studying the dopaminergic system, how they are developed and how their cell fate is decided. And based on this work, a lot of important factors have been identified, such as key transcription factors and key signaling molecules, such as Sony Hedgehog, Wnt1, F8. So since I joined Cornell in 1989, 33 years ago, I initiated the study of the transcriptional regulation of the dopaminergic system using the tyrosine hydroxylase, the first enzyme, as the model system. And then I relocated to McLean Hospital in 1999, which is 22 years ago. And at the time, I started to think about how I can translate my research to treat Parkinson's disease. So initially, many people tried the transplantation of the dopamine cell from aborted tissue. And these are some examples of the papers. As you can see, initially, this cell therapy using the fetal cell were published in top-tier journals such as Science and many New England Journal of Medicine papers, Nature Neuroscience and JAMA, etc. So people were very, very encouraged. I'm just saying some of them, so I think there are hundreds of papers in many other journals. In particular, there are case reports showing that in some patients, the recovery lasts more than 1 or 2 decades, as shown by PET and UPDSR score. So then based on these very promising results, there are 2 groups who performed a big scale double-blind study using many patients. Unfortunately, however, in these 2 double-blind control study, the benefit was not significant. And all the more, in about 25% patients, there were graft-induced dyskinesia. So basically freezing any further fetal cell transplantation, and it was recommended, not recommended as the cell therapy. So in short, fetal cell transplantation study demonstrated proof of principle of the cell therapy in Parkinson's disease. However, fetal cell transplantation has a significant ethical, practical, and medical limitations to become general therapy. So the big question is, what could be the ideal cell source for Parkinson's disease? As I said, many people try to use the aborted fetuses, and many groups are testing adult stem cells. And at this point, the most popular cell source is the dopamine cell from human embryonic stem cell. And then also many people are interested in direct converted dopamine cells. And we all know that the nuclear transfer embryonic stem cell was very hot some time ago. But we thought that induced pluripotent stem cells could be a very promising cell source for cell therapy. As you all know, in 2006, we are all excited that by the Professor Yamanaka's work. He published a groundbreaking work showing that fully differentiated cells like skin cells can be reprogrammed all the way back to the embryonic stem cell-like cells, so-called iPS cells, which ignited the explosive interest both from scientific and general public, because this technology will allow patient-specific or personalized autologous cell therapy. So indeed, these iPS cells revolutionized the whole stem cell research, because it can produce the patient-specific cells, also disease-specific cells, and it can be used for the cell therapy and developmental studies, cellular studies, and more translational studies such as toxicology studies, and also be used for the drug screening. However, as written in this Nature essay, iPS cells were supposed to start the medical revolution, but 10 years after the discovery, they are transforming biological research instead, which means that iPS cells are mostly used for the research, but not the therapy. So we wondered what steps are really essential to realize the potential of the iPS cells for cell therapy of Parkinson's disease. Then we summarized the key issues. The number one issue is a safety issue, because without safety, you will never get the FDA approval. And then second most important thing is reprogramming technology. Although Yamanaka invented it, still our technology to generate iPS cells is still premature, and the mechanisms are very poorly understood. And then number three issue is how to make functional dopamine neurons. So in vitro differentiation is very important. And then we have to do the in vivo safety and efficacy test using the validated animal models. And then we have to do the neurosurgical optimization. And then we need the FDA approval and the regulatory and the clinical issues. So I'll briefly discuss each step, how we addressed these critical issues. The number one issue is safety, obviously. And when we talk about safety, there are two important parameters. The first one is the iPS cells chromosomal DNA should contain the genome integrity. So there should be a normal karyotype. There should be no host chromosomal disruption. There should be no residual DNAs, and there should be no. So the first issue, important issue, is safety. So we have to make sure that the genomic integrity is maintained. And then also, because any single iPS cells can generate tumor, we have to completely eliminate any remaining undifferentiated cells. So to achieve that, we carefully analyzed the whole genome sequencing of the iPS clone we generated using the reprogramming method I will introduce soon. So our iPS lines compared to other known human iPS lines or human ESL lines, the mutational burden was much smaller, showing that the genomic integrity is well maintained. And then we collaborated with Professor Cha in Seoul National University, and we found out that there is a very specific anti-apoptotic gene, which are only expressed in undifferentiated cells. And when we use the inhibitor of that protein survivin, we can selectively eliminate undifferentiated cells. So without the sorting, the tumors can form. But when we use the inhibitor of the survivin and remove the undifferentiated cells, the transplantation gives no tumor and a very safe transplantation. So then we tested whether our chemical treatment do not affect other cells, such as neural precursor cells, and found that when we treat the chemical inhibitor to the undifferentiated cell, it can remove the cell with the efficiency more than 99.99%. But when we treated this chemical to the partially differentiated cell, they did not affect at all. So then when we calculated the remaining undifferentiated cells, when we treated the quercetin, and after the differentiation for 28 days, the remaining undifferentiated cell was basically zero. So showing that our chemical method can guarantee that there is no remaining undifferentiated cells in our protocol. Then second important issue is reprogramming technology. And as I mentioned, although Yamanaka's protocol is very, very reliable to make the iPSC, we found that the efficiency is still not very high, and the quality of iPSCs are not very high. So we then focused on very interesting phenomenon, which is called metabolic reprogramming, also called Warburg-like effect, which was found 100 years ago by the German chemist, Dr. Warburg, who also got the Nobel Prize in 1931 for this finding, which means that he found that when normal cells become cancer cells, their metabolism is totally switching from mitochondrial respiration to the glycolysis. So glycolysis is increased, oxidative phosphorylation is decreased. So this is called metabolic reprogramming. And the underlying mechanism was very unclear. And then we found that the specific microRNA 200C decrease the expression of the gene SIR2. And then when SIR2 is down, it acetylates the key enzymes of the glycolysis. And as a result, glycolysis is enhanced, and it induced the metabolic reprogramming, and then iPS can be generated. So using this technology, we further identified additional microRNAs which can produce these metabolic switches. And then we combine them with the Yamanaka factors, and we determined that when we combine Yamanaka 4 factors with the microRNA 200C and the microRNA 302, we can generate very high quality iPS cells with a much higher efficiency. So we then developed these episomal vectors for the reprogramming process. Then number 3 is the in vitro differentiation optimization. And there are many groups who developed the similar in vitro differentiation method, which was pioneered by Dr. Lawrence Studer and also Jun Takahashi in Japan, Marlin Palmer in Sweden, and Suchun Zhang in USA. So many people use the slightly different but similar differentiation protocol. But then we realized that when you use the differentiation using the monolayer culture, there are many cells who are detached and lost. As a result, the production of dopamine neurons is very unreliable. So we invented a new technology called spotting-based method. Basically, in this case, instead of the whole monolayer cell culture, we define specific spot and code that area, and then let the cells grow and differentiate in that area. So compared to the monolayer culture, here the cells, iPS cells, grow and differentiate in this restricted area. And when we use this spotting-based method, we found that the cell yield is good and cell loss is much less. And most importantly, the cells are much healthier. Then when we use this spotting-based method and differentiate dopamine cells, the resulting dopamine cells show very typical dopamine neuron type in terms of the gene expression and dopamine production and electrophysiology properties. So using that, those dopamine cells, we tested the in vivo safety and efficacy in the NOD SCID mouse. When we transplanted undifferentiated cells, there are 100% tumor formation. When we differentiate day 14 differentiation, no tumor formation, but there are sometimes this kind of outgrowth. When you use the day 28, the graft were very nicely formed and no rosette formation. So we decided to use day 28 cells for the transplantation. When we transplanted the day 28 cells, either fresh cell from the human ES cell or human iPS cell, this is the very popular 6-hydroxydopamine lesion rat model. So without the cell, they after 6-hydroxydopamine, they show the one side rotation because one side dopamine is destroyed and the other side is intact. So when we add the amphetamine, the mouse, the rat shows the rotation behavior. But when we treated the dopamine cell from human ES or human iPS, after 4 months, the rotation behavior was completely rescued. And at 6 months, it showed even like opposite rotation. So we got this complete recovery of the rotation behavior. And when we analyzed the graft, they showed the very extensive reinnervation to the striatum, and then all dopaminergic markers and human markers. Then another important issue is that when we use the autologous cell therapy, these cells cannot be rejected without immune rejection. To test that, we developed the humanized mouse model by using the patient's blood and the purified PBMC, and then reconstitute the mouse immune system with the human immune system, and we generate the humanized mouse. So when we use this humanized mouse, and then transplant the patient's dopamine cell, they are all well received. But when you use the allogenic different dopamine cell in this humanized mouse, they are all rejected without any exception. So proving that patient-derived dopamine cells are not rejected, but the allogenic different source of dopamine cells are all rejected. So then we also optimized the neurosurgical procedure by developing this columnar injection. So using this columnar injection, we can make this columnar shaped graft instead of the disruptive bolus injection, and which can increase the survival of the dopaminergic neurons after transplantation. So using these techniques, we applied the single extended patient treatment to FDA, extended access IND approval. And we got, after many amendments and the renewal, revise, we finally got the full FDA approval to treat a single patient in 2017 January. And we did the surgery 2 times, one side in 2017 September, and the second surgery in the other side in 2018 March, second surgery, based on the FDA's recommendation. So this is a brief summary of the snapshot of the first surgery at the Cornell Medical School, second surgery at the MGH. And this is the, this is the PET imaging and MRI scan showing that the near the surgery time, the PET scan data shows that image is very decreasing, but after the surgery, it's increased, improved, and got very stable. And UPDSR score, the part 3 is partially modestly improving. The decreasing number means increase. And then part 2 and part 1 are very robustly improving. So we published this result last year in NEJM, and which was highlighted in a couple of papers. So in summary, we show that iPS-based personalized or autologous cell therapy is possible, feasible, but we obviously realized that we need a lot more study. And our work was published in these journals and highlighted in some places. But basically, our idea is that from patient, we get the biopsy, like a skin biopsy or blood cell, and we use our optimized method to make the, the clinical grade iPS cell, and then differentiate them to dopamine cell or dopamine progenitor cell, and then transplant to the patients. So I think there are many ways to do this cell therapy. So one way is, as Dr. Lawrence Tudor and some other groups are doing, many people try to use the human embryonic stem cell, but then produce the dopamine cell, and then treat many PD patients using the same batch of cells. So in this case, we need immunosuppression. Another way is to get the iPS cell and the dopamine progenitor cell, and then treat the PD patients with immunosuppression. And another more sophisticated way is to get the human iPS cell and then treat the PD patients with the HLA matching situation. So this is what Professor Yamanaka and the Jun Takashi's group are now currently doing. And in contrast, in our case, we use the autologous therapy. We get the biopsy and the human iPS cell and dopamine cell, and then implant it to the patient's brain. So lessons and the future studies. Can you say that we proved the safety and efficacy of autologous cell therapy? Of course not. This N of 1 study is not enough, but I think we still think that this N of 1 study is meaningful. And I like the verse to share with you these verses, which is in Talmud, which says that whoever saves one life saves the world entire, which was really, I was very happy about this verse. And also I like to this verse, what will it benefit a man if he gains the whole world and loses his own life, which is in Bible. When the patient came to me right before the surgery, he grasped my hands and said to me that, Kwang-Soo, I am dying. So his condition was really worsening at the time. And after this surgery, his worsening stopped and improved, and his condition is very stable until now. now is almost 4 years after the first surgery. So based on that, we are planning to submit an IND application for phase 1 and 2 clinical trial. And the biggest issue is so far this autologous cell therapy is very expensive. So how we can keep the cost down. And I'm cautiously optimistic about this, especially considering the cost of the human whole genome sequencing, which initially 20 years ago took more than hundreds of dollars to do the whole genome sequencing. But after 10 years, in 2010, the price became just only several thousand. And right now it's basically a couple of thousand. So with that, I'd like to acknowledge very talented and hardworking students and the postdocs in my lab. And of course, I'd like to thank our great clinical investigators here at MGH, and also other collaborators at McLean. And I'd like to thank Dr. Kong, who is the bioinformatics specialist and GMP facility, and the clinical investigators at the Cornell Medical School, and Dr. Cha at the Seoul National University. And I'd like to thank these funding sources. And in particular, I'd like to thank the patient who also partly supported our research and volunteered to become the first patient. So I'll stop here, and then I'll talk about if you have any questions. Thank you very much.